Good evening. I'm Catherine Morrison, and I'm the Adult Services Manager here at the Scarborough Library. Again, I apologize. We had a little glitch there starting the meeting, so thank you for sticking with us and logging in tonight. We are going to present our talk, Striped Bass in Scarborough Marsh, the Gulf of Maine and Beyond. This program is co-sponsored by the Friends of Scarborough Marsh. We thank the organization's board for reaching out to suggest this event and for their help in publicizing it. And they certainly did a good job because we had a big crowd sign up for tonight's program. Our presenter tonight is Zachary Whitener, Research Associate at the Gulf of Maine Research Institute, or GMRI, in Portland. Zach's research focuses primarily on fish populations, including age, growth, and stock structure. Zach received his bachelor's in anthropology from Brown University, where his studies focused on marine resource management. He later went on to receive his master's in marine and environmental sciences from the University of the Virgin Islands. Outside of GMRI, Zach is a registered Maine guide and U.S. Coast Guard licensed captain who owns and operates his own company, Anadromous Adventures Guide Service, LLC, which specializes in fly fishing for striped bass in Casco Bay. He has a passion for fishing, cooking, and if lucky, cooking fish. So please help me welcome virtually Zach to the library this evening.
Great. Thank you so much. It's wonderful to be, I guess, in my office tonight, but virtually in the library. Thank you so much for the nice introduction. I wish I could see you all in person, or if I could see more of your faces to see how things are going. Well, I'm going to do my best. As was said, I am both a scientist at GMRI and I'm also a Maine guide. So tonight is a kind of a fun thing for me where I get to wear two hats. I get to kind of switch back and forth between scientist and fishing guide and tell you what I think that you should know about striped bass around here. So as was said, I'm going to an outline for my talk. I'm going to talk first about striped bass, just in general, their life history, the human history, our interactions with them. I'm then going to talk about Scarborough Marsh and other estuaries, the ecology. That's what we're going to talk about then, fishing strategies and things too. And then I'm going to finish up with the current status of the fishery and research, some of the research that we're doing and how you can get involved if you're interested. So part one, what are we talking about? So we're talking about Moroni saxatilis. So basically that's Latin for rock bass. Down south, stripers get called rockfish because of their association with hard bottom, wedges, rock piles, things like that. Then you come up to Maine, unlike in the Chesapeake, and everything's a rock pile. So it doesn't make much sense to call them rockfish up here. Everything's a rockfish. Commonly, the size that we see around here are, I'd say, between 14 and 30 inches. I wish we saw them bigger, but we do on occasion, but most of them are what are commonly referred to as schoolies. So smaller fish that hang out in schools together. Stripers eat everything, which is really convenient if you are someone who's trying to catch them fishing. They readily will take bait, flies, lures, all sorts of ways to fish for stripers. As you can see from this picture, I'm sure most of you are familiar with the striped bass, but as you can see from this picture, they're shaped like a typical bass. Moroni, the genus, is the same that as white perch, actually. And in some parts of the range in the Chesapeake, they hybridize. So they're very closely related to perch. And because of that, or part of that, you can see the spines across the top of the dorsal fin that will definitely give you a good jab if you're not careful with them. As you can see in this picture, people wearing winter hats fishing. People are obsessed about this fish. The southern part of their range, there's a number of game fish or fish that are caught recreationally, such as bluefish, fluke, etc. In Maine, striped bass are really the only game in town for saltwater fish. We used to get a lot more bluefish around, and that doesn't happen as much anymore. People, of course, we all still fish for mackerel, but stripers are really what drives the recreational fishing industry on the coast of Maine. They are just starting to show up now. I've heard some rumors. I haven't been out myself yet, but that's a project for later this week. So seeing people wearing winter hats fishing on the beach is something that you'll probably see this week if you haven't yet. I like to fish for them from a boat. You can have all sorts of your friends with you at the same time. That being said, of course, people fish from the shore, especially in places like Scarborough Marsh, with grassy banks. It's easy to walk around and gives you access to fish. But we'll talk a lot more about the fishing later on. So the big question is, that I'm going to talk a lot about tonight, is the stripers we have in Maine, from whence do they come? And so the general population of stripers on the East Coast, most of them spawn in the Chesapeake this time, or March, April. And then the Chesapeake starts to get too warm for the bigger fish. They like cooler water. So they'll start to move north along the eastern seaboard, eventually come up to Maine for the summer, and then come south in the fall, and then back down to the mid-Atlantic to overwinter. That's probably 90% or more of the fish that we have in Maine are actually migratory fish that come from the south. And so during the wintertime, there are very few striped bass around here. There are still some, but we'll talk that a little bit later. And because of these migrations, and because there's such a popular recreational fish, magazines and websites like onthewater.com have their weekly striper migration map, where they will let you know what size fish and where they are so far in the migration. Generally, the smaller fish migrate first, and then the bigger fish, once they're done spawning in the Chesapeake, which is home to probably 80 to 90% of the spawning. Once they're done spawning, they start moving north. But where else are they? They're not just, as I've mentioned, most of the fish we have around here are migratory, but it's not all of them. Actually, the native range of the fish, according to USGS, goes all the way from the Gulf of Mexico into the Bay of Fundy. Well, as this is a US government website, oftentimes it happens with fish and other species, or they think that everything stops at international borders, when in fact they don't. Fish don't know where borders are. So actually, there's a separate population of striped bass that spend most of their lives in the Gulf of St. Lawrence. And so there's spawning populations in the St. Lawrence River. The Gulf of St. Lawrence population has really exploded in the last few years, and there's been some competition with Atlantic salmon. But the fact is, those species have co-evolved since the last ice age, since this habitat became available to them, to all inhabit the same waters and rivers. And then this map also shows the Bay of Fundy population, or they're spawning in the Bay of Fundy, them coming along the coast of Maine. What's interesting about this is that as you move further north and south, the population and the way the fish migrate aren't what we're used to on the East Coast. And so oftentimes the northern fish and the southern fish, say south of North Carolina, spend a lot more time in rivers throughout the year. So what are they doing in all these rivers? Are stripers like salmon? Yeah, sure, sort of. Oftentimes they're found in the same rivers in Atlantic Canada. Generally speaking, they're both anadromous, which means they're fish that spend their adult life at sea and return to the freshwater where they were spawned and hatched themselves to spawn. And so just like how salmon famously go back to their own rivers, striped bass do too, for the most part. So like I mentioned, both species have complex life histories that can be a lot more interesting than we normally think about with different behaviors, such as going up rivers and down rivers at different times. But the big thing I want you to take away from this is that two things drive migratory behavior. That's spawning and feeding. And so sometimes the areas that are really good for spawning for fish, like the Chesapeake Bay, that has the ideal conditions for spawning and hatching of fish, and what we as fishery scientists call recruitment. It gets too hot for the bass in the summertime, and most of the big fish will leave. And so they leave to go someplace where they can eat. And that's what brings them to Maine. By the time they come to Maine, this time of year in May and June, they are hungry. And this is the time of year to get out there and go fish for them. The criteria for successful spawning, there's a couple of different studies that basically it's between 12 degrees and 20 degrees centigrade, which is a really big range, but it shows that the fish have adapted to different estuaries. In general, though, the specific gravity of the eggs are slightly negatively buoyant. And so that's why rain has a big effect on striped bass recruitment in the Chesapeake. Basically, the salinity of the water controls how much the egg, when it's freshwater versus salt, controls how much the eggs float or sink. If they sink too fast, they'll get covered up on the bottom. If they don't float at all, they'll get eaten by other things. And so there really has to be this perfect combination of temperature, salinity, and flow of the river. So that's why striped bass have really wild swings in recruitment, as we fishery scientists call basically the number of fish, the number of eggs that become adult fish, or at least juvenile fish, and how successful they are year to year. And so recruitment is really based on environmental factors. So where are the other places where striped bass spawn besides the Chesapeake and the Delaware? Well, you may know that there's some that spawn actually in our own backyard in Mary Meeting Bay, which is if you don't know about Mary Meeting Bay, I won't go into it. It's going to take forever because I'm really excited about it. Mary Meeting Bay is amazing. It's a tidal freshwater ecosystem. It's an estuary about 20 miles inland up the Kennebec from the ocean, but it has spawning striped bass, which I'll get a little more detail about in a minute. But there's also fish that spawn in the Bay of Fundy. And so we think that some of those fish in the Bay of Fundy, probably the juveniles, probably come down to the Gulf of Maine on occasion too. What's really cool about the Bay of Fundy populations, like I was saying before, they exhibit different life histories. Some of the fish don't do this big migration like we're used to on the East Coast. In the Shubenacadie River, I'm sorry if I've mispronounced that, I'm not Nova Scotian, but in the Shubenacadie River, what's really neat is that their migration is basically opposite from what we're used to on the East Coast. And so by that, I mean, the fish actually spend the winter in Shubenacadie Lake, in a freshwater lake where people catch them ice fishing. In the springtime, those fish go out to the ocean to feed. And so their big run for fish is in the springtime when fish are going downriver, which is completely backwards to what we on the East Coast of the US are used to. Then in the fall, they come back up those rivers after feeding in the Minas Basin, the Bay of Fundy, to overwinter in the lake again. It's a super interesting and to us bizarre life history that is so backwards, but it's really neat to think about all the ways that these fish probably used to live, all these different ways up and down the East Coast. Because historically, there were striped bass in every major river on the East Coast of the United States. I'm just going to show you a quick video of spawning bass, because this is what you would see. And if you see this in Mary Meeting Bay, let us know. So this is what spawning striped bass look like. So this is the end of May in 2018 in the Miramichi. And so it just looks like they're splashing on the surface. And so those are males and females releasing gametes to fertilize the eggs. And this whole river is full of fish. In the last few years, the population has almost tripled for this population, and we're not really sure why, but it's made for some incredible fishing conditions. And so just seeing something like this, see all the boats in the background here, these are all people fishing. It's become a huge, huge fishery in Nova Scotia for these fish, or sorry, in New Brunswick. So, I talked about how we had them, that they were up and down the East Coast everywhere, all these major rivers and things. So where did they go? Well, a lot of that is from habitat degradation, whether that and loss of access to spawning areas, whether that's dams or the cutting down of the forest. Log drives are very detrimental, both to striped bass and to salmon, that luckily don't happen anymore, but that creates a ton of erosion and physically pushes fish out of their spawning areas and makes for terrible water quality. And then in 1837, the big one in Maine was the building of Edwards Dam on the Kennebec River. And so this basically cut off most of the spawning area for striped bass in the Kennebec River. And of course, overfishing had a really big impact on bass, including commercial fishing. And so one of the things that I really enjoy reading is historical documents to think about and to see what fisheries were like before industrialization. And so early authors, sorry, there's gonna be a lot of words here, but early authors described the striped bass as coming up the rivers at alewife time, which was, it's right now, it's May. So this is from a 1634 report on the fishes of Massachusetts. And talking about striped bass coming up to the Charles River, the Saugus, the Chevico, the Parker, Great Bay, Piscataqua, the Mousam. If you know these rivers, the Kennebec, the Penobscot, it's amazing to think that each one of these rivers had its own unique spawning population until basically the Europeans came over and destroyed them. And so it's amazing to think about how much more productive this fishery had to have been with so many different spawning areas. And I talked earlier about the wild swings and recruitment from the Kennebec. What happens when you have different spawning areas is that some years might be bad for some places, but better in others. And so by having only a few major spawning areas left now for striped bass, we're really dependent on basically the weather in the spring in the Chesapeake for the rest of this fishery up and down the East Coast. And so it's amazing to think about what we could have if some of these populations were able to reestablish themselves. Again, some of these historical, again, from Wood of 1634, on the smaller estuaries, the striped bass would tide in and out, which meant that they came in and out with the tide. And the English at the top of the high tide would cross the creeks with long seines or bass nets, which would stop the fish. And so this is what's called a stop seine. And so basically, as the tide comes in, the fish come in, they put a net across the bay, the tide goes out, you've got all your fish there on the beach. So this is the part that's wild to me, is these numbers of fisheries of spawning bass in Maine, not that long ago. So on the Sheepsket, an estimated catch of 5,000 pounds in 1887, 27,000 pounds, the entire coast of Maine. The fisheries in decline. By 1902, only 1,600 pounds were caught, and then by 1919, 600 pounds. Other rivers in eastern Maine with substantial fisheries, the Penobscot and the Androscoggin, which lost its run as early as 1860. For river after river, I found mention of the year that the striped bass runs ended as the fishery destroyed itself. So these fishermen knew what was happening if they're fishing for spawning fish at their spawning time, and each year they're watching their catch go down, and it's really a shame that this happened. But it does give hope to me that someday some of these populations could rebuild themselves. The last evidence for spawning in the Gulf of Maine was in the Mowsom River down in Kennebunk in 1937 when juveniles were caught, and the Parker River in 1953. After that, spawning was considered, the spawning bass are considered extirpated from northern New England north of the Hudson River. So in 1982, the Maine Department of Marine Resources started a restoration effort to bring striped bass back to Maine. And so from the 80s to the early 90s, they took juvenile fish, so fingerlings that had just been hatched from the Hudson River, and put them in the Kennebec River. And we do this, our agencies do this with juvenile fish that are anadromous, because even though those fish weren't hatched in the Kennebec River, they implanted on the Kennebec. So they spent enough time as these little tiny juveniles, basically to think of that as home. So after a number of years of stocking young of the year fish, eventually we reestablished a spawning stock in the Kennebec. It's very small, but it's still there. And so these fish still get caught on occasion from the spawners. That's why we have special regulations on the Kennebec, because it's to protect spawning fish. And later in the summer, when the Department of Resources does surveys on the Kennebec, they'll catch these fingerling young of the year striped bass. So that's my quick overview of striped bass ecology and life history. I would like to take a couple minutes for questions if you have any right now.
Zach, we do have a question here. Let's see. It says, you said that salmon and striped bass can compete near the border. How do they interact or compete? Are they territorial, aggressive, or is it just competing for food?
Well, it's both. And so what happens is striped bass, like I mentioned earlier, will eat everything that'll fit in its mouth. And unfortunately, salmon smolt, which are juvenile salmon, would, unlike striped bass that only spend a little bit of time in their natal estuaries before going out to the ocean and start rapidly growing, Atlantic salmon will spend a few years in the rivers that they were spawned in before they go out to the ocean, get much bigger. And so when those Atlantic salmon smolts are in the river, they are very tasty snacks for striped bass. And so there's a lot of contention right now on these historic salmon rivers in Atlantic Canada, where now there's the exploding population of striped bass. The salmon fisheries have been declining because of the same things of habitat destruction, dams, overfishing. And so these salmon fisheries are really struggling. And now they've got a much bigger population of predators right in their home. So it's a really tough balancing act that Canada is struggling right now with. How do we protect our native striped bass that we also brought back from the brink of extinction? But how do we manage those without hurting our Atlantic salmon, which are headed towards extinction themselves? So it's a really tricky situation.
Okay, and here's a question from Peter. There's talk of removing the 4 major dams on the Kennebec for restoration purposes, Lockwood Dam all the way to Madison. How far upstream would stripers potentially migrate? Would it follow the Alewife Run?
I don't know that exactly. I haven't seen in the historical documents how far up they'll go. They are strong swimmers, but in general, they generally don't jump. And so they typically won't go past significant natural falls the way that salmon can. So some years, striped bass get into ponds in Maine, like Seven Tree Pond, because they follow the alewives all the way upriver and get into a pond. And so they'll follow the striped bass, they'll follow the alewives, one of their favorite foods. A good distance upstream, but I don't know how far historically they used to go. That being said, with those dams removed, it'll greatly expand the range for the alewives and other river herring, which makes a huge forage base for striped bass and everything else that eats alewives, which is pretty much everything that eats fish in the Gulf of Maine.
Okay, and here's a question from Michael. Recently, the dam in Westbrook was removed and a fish ladder installed. Do you have any migration data on stripers in the Prisumscot River at Westbrook?
Not at Westbrook per se. We've done work over the years to see for the Millbrook run of alewives. So the alewives come up Prisumscot Falls, where Smelt Hill Dam used to be, and they go about, I think, probably 4 or 5 miles upstream, then turn right to go up to Highland Lake. Overall, it's about a 7-mile run to the lake for the alewives. And so we've done a lot of sampling at the old Smelt Hill Dam at Prisumscot Falls, and we've caught stripers there while sampling alewives. But I've never heard of anyone catching them upstream from the falls. So that doesn't mean that they never get up there, but even anecdotally, I don't know about them ever going past those falls. Again, because they don't usually jump up falls. That being said, they could. And similarly with, like I was just saying about the Kennebec, with the main stem of Prisumscot, more of it open thanks to the dam removals and fishways in Westbrook, that should greatly expand the blueback herring population, which will attract more predatory fish into the Prisumscot. So they very well could go up it on occasion. Nope, you're muted.
Sorry. Evelyn is asking, do stripers compete with shad for food and territory?
So stripers will definitely eat juvenile shad. So once the shad spawn in the spring, if the young of the year are up there, they will eat them just as fast as any other river herring. One of the funny, just a quick anecdote about fishing in the Spurwink Marsh, so one over from Scarborough Marsh. I was there one September day, and I found a little eddy where I was casting a fly into it, and on successive casts, I caught a 12-inch striper, so a little tiny schoolie. The next cast, I caught a 12-inch shad that was up there fishing, so on the same fly. And then the next cast was a striper, and then a little bit later, I caught another shad. And so at the same size and the same place, those fish probably were competing for food. If I was catching them on the same fly in the exact same place, but again, at different times in their lives, the stripers could be eating the shad. So I think that, similar to salmon, they all, for lack of better terminology, grew up in the river and they evolved in these rivers together. And so they definitely have competitive relationships, but the relationships have been around as long as the fish have. So, and I'm going to get going on the next section here. Because like I said, I could talk about this for days, and I've got too many slides.
Okay.
All right, so into the meat of the presentation, I'm going to talk now about Scarborough Marsh and other estuaries, and some fishing tactics associated with that. So like I mentioned earlier, migration behavior is driven by two things, food and spawning. Since we mentioned that there's no spawning in Scarborough Marsh, that means the striped bass are there to eat. What makes estuaries special? So estuaries are flooded coastal wetlands, freshwater inputs, and saltwater, lots of nutrients, tidal mixing, currents, everything mixed around. There's 5 rivers in Scarborough that come together to make up Scarborough Marsh. Scarborough Marsh is about 3,000 or 3,500 acres it's a really special ecological place that we have in southern Maine, that we're so lucky that the state and other people were smart enough to protect it before it could all get filled in. And so with the average 9-foot tides we have here, it makes a really complex bathymetry, topography, hydrology. So water swirling around all the time. You'll see a whirlpool at one place, and 10 minutes later, it's not there. There's so much going on with water moving around. Again, that mixes up nutrients, which brings in more bait fish, which brings in predatory fish, also birds, all sorts of things. It's great for everything. It's a soup of life, really. The other cool thing about estuaries is that they're warm. And so generally muddy or sandy bottom, shallow, they warm up a lot faster than the rest of our coastal ocean. So striped bass like warm water to a point. And so basically what we say this time of year is that the bass won't show up until the ocean water is about 50 degrees. And that's when they feed very well between 50, I would say 55 and maybe 65 or so degrees, in my experience anyway. And what happens though is the ocean right now, the other day I was out in my boat in Casco Bay and the water was 54 degrees in the Royal River and 44 degrees out by Long Island in Casco Bay. And so what the bass do in the springtime, a lot of times they hopscotch up the coast. And so they'll be in a warm water river eating in an estuary in a marsh, and then they'll go up basically to the next river because they'll tolerate that cold ocean water for a little bit, and then they go back into the rivers where it's warmer. And so early season up here, so May, June, these shallow marshes are great for fishing because they're warmer, which means the bass are feeding more in addition to all the food they have available too. In the obligatory high tide, low tide picture. So the really cool thing about salt marshes is their primary productivity. So you see all this bright green, now they're just starting to get green again, this bright green marsh grasses, the Spartina grass. It's among the highest primary productivity of any ecosystem in the world, these salt marshes. They're dominated by different species of Spartina. Of course, there's other grass species. But what's really important is all this mud and detritus. So dying things and things eating them, benthic macroalgae. There's microbes in the mud. You know, mud is not dead. Mud is very, very busy. And so there's a lot of nutrient cycling that's going on. But at the same time, you've got these strong currents driven by the tides and the river flows that are constantly scouring and mixing things up and making all these nutrients available for other primary producers, then secondary consumers, et cetera. Salt marshes are, they're almost like the habitat version of the alewife for me. Alewives are my favorite fish. They get eaten by everything. They're really important. You can't explain how important alewives are. It's the same thing with salt marshes. They're incredibly important ecologically in this area. And we're very lucky to have Scarborough Marsh right next door. Of course, all this primary productivity and primary and secondary consumption leads to an explosion of animal life, birds, invertebrates, fishes. I had to throw a glossy ibis in here. The first time I saw one was in Scarborough Marsh, and I was blown away. This was about 10 years ago. I grew up in Casco Bay, just off of Portland here. We don't have any ibises in Casco Bay. So the first time I saw one of these, I was blown away. I just had to throw in my favorite salt marsh bird. So anyway, as a result too of the invertebrates, also makes Scarborough Marsh and other marshes an important place for clamming and other fisheries uses. You know who also likes clams? Striped bass, as you bait fishermen definitely know. So two things I said earlier, two things make fish migrate: food and spawning. And then similarly, two things concentrate fish. So what help you, things to think about when trying to figure out where to fish, the things that really bring fish together are currents and food. And if you're not, if you don't know what's on the menu, you are the menu. That's definitely the mantra for life in a salt marsh. Everything is eating each other all the time. So we'll talk more about currents and food here. So I'm going to start at the Upper Marsh, which is, so think about it like up near Route 1, the upper part. It's often really steep banks. These floodplains that only flood generally on spring tides. They fill up the salt pans that oftentimes seem like they're full of stagnant water, but there's a lot of life in there, including mummichogs and other little fish. So you'll get currents through here. A lot of times, though, it's really hit or miss for fishing, depending on what's going on. There's not a whole lot of places for the bass to hold. And by that, I mean places to sit in the current and things like that. And so a lot of times they'll come in and out with the tide in spots like this. This is just an aerial view of the marsh. You can see all these sinuous channels all over the place. I mentioned the mummichog, which Fundulus heteroclitus. These are the little minnows that you see in the salt pans. In one of those salt pans that's maybe 10 feet in diameter and a foot deep, plus 3 feet of muck, there might be 200 mummichogs in that salt pan. They're really important fish that are found up and down the East Coast. And everything likes to eat them. They can get their mouths around them, their beaks on them, what have you. Of course, you have to have mummichog fly. I'm going to talk about fly fishing because I'm a fly fisherman primarily. And so I tie flies to match these. Similarly, Atlantic silversides are very common in the Upper Marsh. They're pretty common everywhere in the marshes. They're more active in the marsh in summertime. They spawn in June, a lot of times June and early July. If you catch one that time of year, you see a super distended belly that's full of eggs. And they gather in larger schools later in the season with birds and bass and everything trying to eat them. So again, a lot of fly fishing is mimicking what you see for natural bait. And so part of, like I said, currents, you have these currents in the marshes, you can follow the current seams, but the other big thing is looking for food. And so if you see if you're walking up to a salt marsh edge, one of these grassy banks and wondering where to fish, walk over near where the birds are, because the birds don't just stand around for fun. They're right there fishing themselves for sticklebacks, mummichogs, whatever else is around. And so you know that if there's bait fish around, there's probably some big fish around. One of the most exciting times to fish in the salt marsh is at high tide. There's a lot more room, volume-wise, for fish to move around. And so there's a lot more room for things to get concentrated corralled by fish, by bass. So you come up on a bank like this, nice glassy calm water, and this is my best effort to animate things. And you'll start to see some ripple on the water. And so you see there's no wind, it's nice and flat calm. You'll start to see these ripples. And oftentimes those are predatory fish like striped bass moving along the edge of a bank. And then all of a sudden you'll see the bait fish, whether it's mummichogs, silversides, what have you, that splash up out of the water. And so you can see this, from a quarter mile away as you're walking along or in a boat, making your way up the marsh, and you can see immediately, okay, there's fish, there's food, that's a place to go fish over there. The other thing is reading the currents in a marsh. What can really help is seeing where's sediment gets deposited. That can help you read where the water flows and how it flows. And so you see these blue lines coming down. This is the general flow of this river on an outgoing tide. You can see these big sandbanks here. So this is where obviously water is slowed down enough for that suspended sediment to drop out of the flow. And so at high tide, what you'll have here is an eddy where water is now curling back on itself. And stripers are fast and voracious and lazy. Like any good predator, they're not going to work any harder than they have to. And so if they can find a place where they can sit and have food come to them, they will. And so oftentimes at a place like this, I would fish standing on this bank and cast out into the river and have my fly or lure swept with a natural current back into this eddy, because oftentimes where fish will be is either sitting over here facing downstream, because in reality the current's coming back towards them, or they'll be along this edge here where the water splits off from the two different currents. And so by reading the water and seeing where things are flowing and how they're flowing, you can have an idea of where the predatory fish might be. But it's not just on a river scale. These big flows, or this river is 100 yards wide here, but also on really small scales. I talked for a moment about how lucky we are, the condition that Scarborough Marsh is in. One of the things that didn't happen very much at Scarborough Marsh that happened in the salt marshes south of us are ditching, where people cut these channels, these ditches through the salt marsh to drain them, so they wouldn't hold water at high tide. It's for mosquito control and to make it easier to hay on the salt marsh and things like that. Ecologically, this is a mess. I'm very glad that Scarborough Marsh looks like this on the right with these more sinuous channels. But that being said, every one of these channels, whether it's a real ditch or these natural sinuous channels, every one of these is a mini river, whether it's 2 feet wide or 40 feet wide. They all have their own currents coming in and out. And where that water swirls, where those current seams are, you have a pretty good guess of where you might be able to find some striped bass feeding. And so as the tides move up and down, and the currents move in and out, the salt marshes are incredibly complex and ever changing. And you can get an idea of how to fish it. And of course, there's some obvious places where there's current. This is on the west side of the marsh, coming out from an old, where there had been a cofferdam. Basically, splashing white water, it's the same as a river or some of these other less obvious currents that fish will hang out kind of on the edges of these seams because it's an easy place for a meal. I love to fish these marshes from a canoe. It's silent to move around, super fun to actually look down and see the fish. You don't scare them anywhere near as easily. Of course, you can also fish them from a boat. But you can also walk the banks like I've been talking about. So there's a lot of different ways to fish these upper marsh areas. I'm now going to talk a little bit about the lower marsh and the transition to full salinity. Much more influenced by salt water. Typically, there's a sandy bottom with larger volumes of water moving. If any of you have ever been to Ferry Rock on an outgoing tide, you know that you want to stay on the rock and not go swimming, because you'll end up at end up over at Black Point if you're not careful. So this is an aerial view of the marsh entrance right there. This is Ferry Rock here, Pine Point over here. These boats are fishing. You can tell because of exactly where they are. They're in the right places where the current comes down each side of the marsh and will break over so you'll get turbulent water, again, mixing up. Bait fish get confused. So that's what these guys are fishing for. And these guys, you'll get eddies off the side of this. edge here. And so fishing on these edges is oftentimes more productive than fishing right in the middle where there's a solid stream of current going. So you want to look for those eddies, current seams, things like that. And of course, like I mentioned earlier, you don't need a boat. Stripers are it's one of the most important things about striped bass is how accessible they are. Whether you're standing on a rock like my wife here, or you're standing on a beach trying to not get swept out from a river, an outgoing tide. Striped bass are so accessible. That's why they're so popular. But one of the things about the lower marsh is there's a much wider variety of food available. So these are sand lance that, as their names imply, they burrow down in the sand to hide from the strongest currents. Then they'll pop up again. Fun to mimic because they're little fish. Atlantic herring, juvenile Atlantic herring come up into the marsh to feed. Atlantic mackerel can be found on the outside edges of the marsh. Sometimes they'll venture up inside. Again, there's a whole variety of flies we use for these. River herring, although they don't, there are some shad that spawn in the Nonsuch River. Typically, there's not much spawning behavior of river herring in Scarborough Marsh. However, it is a feeding area for juvenile fish, for juvenile river herring. So they'll go up there, and even adults, after they've spawned, will then move into Scarborough Marsh or other coastal estuaries to continue to feed, to build up their energy reserves after spawning. And so these bait are around green crabs, which ecologically everyone hates more than anything because they eat the clams, they eat eelgrass. They're an awful invasive species. The good thing is stripers like to eat them, so at least they've got that going for them. So crab flies work especially well, especially in sandy areas like Scarborough Marsh. If you get a little bit further out, pollock are ubiquitous fish around here. Again, your Atlantic silverside, we have smelt in the marsh. That is a giant smelt. Again, stripers will eat anything they can get their mouth on, and they especially like oily fish like mackerel, smelt, alewives, herring. Of course, there's also pogies, which south of us they call them menhaden, which is hard to say. It's not a good word. We call them pogies, we fishermen, otherwise known as bunkers south of here too. They've made a huge comeback in this area in the last 10 years, which is fantastic. It's an amazing amount of life brought inshore. Sometimes not only bass feeding on them, but sometimes you'll see whales right off of Higgins Beach chasing pogies inshore. They're an amazing fish that I'm very glad have come back. We have lots of little grass shrimp in the marsh, American eels, of course. We have squid, especially in the summertime. And of course, different species of annelids. So that means basically many armed worms, many segmented worms. So clam worms, bloodworms, a couple of different things that you may have seen them, what we fishermen call it a worm hatch. It's not really a hatch. It's actually, it's part of their spawning behavior. They segment, depending on moon cycles, a couple of times per year, a segment of the back of the worm will break off and swim to the surface, and all these little bits of worms will be swimming around, and it's spawning behavior, and the fish go bananas for them. So I mentioned briefly about crabs and crab flies. They really do work. This is the Scarborough Marsh, on the edge of the marsh. This is a client I took fishing one day. He didn't believe me that crab flies were a thing, but if you put people on fish, they become believers. I'm going to skip over that, just some of the other flies that we use around here. But the exciting thing, so I told you two things that attract fish, or two ways to help you find fish, is currents and food. The biggest way to find where there's food, unless it's an obvious school of fish getting attacked, it's birds. Birds are the fisherman's best friend. And not only just birds in general, but what species of bird and how it's behaving can help you figure out what the predatory fish are eating. And so if you have a flock of terns, that means, and there's no herring gulls mixed in, typically that means that's very small food. So usually either sand lance or Atlantic herring, juvenile Atlantic herring. If you have herring gulls, a big flock of herring gulls, so these are birds on top of a school of stripers. So the stripers push together the bait fish, they push them to the surface, then they're easy pickings for the birds. And so if you have herring gulls with no terns around, that you know then that the bait is too big for the terns. And so these stripers were feeding on river herring. And so if you find a mix of terns and gulls, that means that the bait is probably small enough for terns, but gulls are interested in it. If you see terns but no, or see gulls and no terns, that means that the bait fish are bigger. Then, of course, the cormorant that you see everywhere right now, especially for alewives, they're big predators of alewives. And so starting weeks before the alewives show up, you'll see hundreds of cormorants lining our rivers and brooks, waiting for those first waves of alewives to come up. And what's fun about cormorants is that you can see what they're eating because they can't swallow the fish until they come up to the surface again. So you get a quick glimpse to see what they've been eating. Whereas when the terns or gulls are diving down, they typically swallow the fish too fast to really see what it is. So that's the neat thing about cormorants. Of course, when you see something like this, as fishermen, we get really attracted to birds and we'll rush to see where the birds are feeding. And looking at our depth sounders, you can see these are actually these marks here, actually striped bass on the bottom that I found because from chasing a flock of birds and coming up over where the birds were, you can see then the predatory fish on the bottom. And then these on the right, these larger, more diffuse marks, that's a mix of bait fish and striped bass. And so it's one of the tools that we use to see what's below us and also the temperature. A lot of times, like as I mentioned earlier, the temperature really affects fish feeding behavior. Like I said before, one of the nice things about having a boat is it makes it a lot easier to chase birds. You can run down the beach chasing terns and hope that the stripers are still pushing bait inshore. But if you're in a boat, you can go chase them, even if they get off the beach. Like I mentioned before, currents, moving water are really important, whether it's the mouth of a marsh, breaking waves on a beach, or breaking waves on rocks. These are all places where you can find adult predatory fish. I also threw in a picture of Back Cove here because, like anything in life, fishing especially, for every rule, there's 1,000 exceptions. And so similar to the high marsh in Scarborough at high tide, Back Cove is actually full of fish. And that's because they can push bait fish basically along the channels in the cove. Even though there's not the sod banks, there's still sinuous channels at low tide throughout the cove. And so it acts as similarly to a salt marsh. I've got a minute. I'll talk about this real quick too. And just kind of the fishing strategy of these places. If you were standing at the X, you want to cast along the rocks, not straight out. Fish aren't straight out. Fish are at your feet. They're in that turbulent moving water. Similarly, if you're on a boat in the same place, again, you want to cast towards the rocks. Bank fishermen are always trying to cast as far as they can out, while at the same time, boat fishermen are always trying to cast as close as they can in. Nobody's ever happy where they are, but one of the important things to remember is that the fish are always going to be moving turbulent water. Okay, so whew, that was a lot for the midsection. I'll take a couple quick questions before I'll run into the final bit, which is going to be about research.
Let me unmute myself. With sea level rise, simulations show that there could be significant salt marsh loss in Scarborough, but large potential gains in mudflats. Could you comment on all about this? Potential conversion might influence stripers and what they feed on.
Yeah, that is something that I am terrified of as somebody who spends an immense amount of time in salt marshes throughout the year. I'm really afraid that this is going to be, I hope it's not the end of them functionally as they are now, because without more, basically the marshes can't grow and deposit sediment fast enough to keep up with sea level rise. And so I think that the fish will adapt. I think they'll find things to eat. Like I said, they eat heavily on green crabs, which is not a food they ever saw until green crabs came over from Europe. And green crabs are very detrimental to salt marshes, but they also live in a lot more habitats. So I think that the fish that we have will adapt to new environments, but that certainly doesn't mean that we should be okay with what's happening. Yeah, the drowning of salt marshes, I find a terrifying proposition.
Then another question: what is your what are your favorite birds to see when you're fishing?
Like I said, the glossy ibis. I love oyster catchers. Oyster catchers are so cool. I've had a love-hate relationship with ospreys. To be honest, the racket they make drives me nuts, but they're really cool to see, and I love to watch them fish. And they remind me a lot of, they remind me of home. And so, as you mentioned earlier, yeah. Oh, sorry. As I mentioned earlier, I went to the University of the Virgin Islands for my graduate studies. And one day I didn't know how far south ospreys migrated. And so the first winter I was living in the Caribbean, I heard this awful racket and I couldn't put my finger on it, what that was. That wasn't there yesterday in the mangrove. And then I realized, I looked up and, oh, I probably know you from Casco Bay. You're probably the one that yells at me when I'm fishing on the beach. And of course, every species of duck. I'm a duck hunter, and so I love watching ducks, hunting ducks. So black ducks are the epitome of our salt marsh species to me. So I love anytime I can see black ducks and watch their fun behavior.
We have a couple of more questions.
Sure.
Can you discuss the current recommendations to decrease mortality from foul hooking or wearing out fish when played too long while bringing them in?
Yeah, we're going to talk a little bit more about the status of the fishery in the next section. But as a quick preview to that, yeah, it's incredibly important to, when you hook a fish, to not tire it out. And so as fly fishermen, we never get to put fish on reels. Is kind of the thing. If you know anything about fly fishing, is that generally the fish are small or smaller trout and things. You can pull them in by hand. It's fun when you get a really big fish to have actually take line off the reel. And so oftentimes when people first start saltwater fly fishing, they get really excited about, oh, this fish is pulling, I'll put it on the reel. And that's not necessary. A fish will tell you when it needs to go on the reel, which means it's really fast, it's really strong. So there's no shame in not wasting your time in pulling in your fish stripping the line. It's very important, especially if you catch a lot of small fish, to crimp your barbs, to be fishing barbless, creates far less damage on the fish, much easier to release them. And if they come off the hook, hey, that just makes your job a lot easier. The fun part is hooking it and fighting it. If it pops off with very minimal contact with you, that's better for everyone. So yeah, I'll talk a little bit more about fishing status of the fishery in the next section.
Okay, this next question you may be addressing in that, so I'll save it
Sure.
for later. Okay.
Great. All right. Well, I'm going to go to that next section then. Again, sorry this is so long. I could talk for days about this. Let's power through together. All right. So now I'm going to talk about the current status of the fishery and our research. So the question that everyone wants to know is, are there a lot of fish? Are there little fish? Are there big fish? What do we have? And what's going on? So in 2019, I guess before you read the top there, look at this graph. This is the spawning stock biomass of female striped bass. And so this is, as fishery scientists, this is the number we're concerned with, which is the number of mature females. And striped bass were not doing well at all through the 1980s. And there was a moratorium where all fishing was stopped for striped bass. The population rebounded and was doing great through the 90s and early 2000s. It's since started to decline. One of the things about fisheries management is that we're always playing catch up. Every year we get new data, and then we use that data to back calculate to basically correct ourselves what we thought was happening then to what we think about was happening now. And so it's really hard to know how well a fishery is doing in an instant, but looking back in time is easier. And so the important thing about this graph is that this blue blob, the female spawning stock biomass, it is below, at all times, it's below the solid green line, which is the target, which is the number of spawning females there should be in the population. And for the last 10 years has also been below the threshold. So the threshold means the absolute minimum that we have to have for a sustainable fishery. And so striped bass is, as of 2019, scientists thought there were 151 million pounds of spawning stock bass when there should be at least 200 million pounds. So that's a problem. So that at that time, striped bass were called overfished. There's two terms we use a lot that seem interchangeable, but they're not: overfished versus overfishing. Overfished is an adjective about the stock. So all overfished means is that there's not as much spawning stock biomass as there should be, below the target and below the threshold. On the flip side of that, or the other thing we talk about, is overfishing. And so basically we calculate that as mortality. I'm not going to go into the statistics about how it's actually calculated and what exactly F means, the fishing mortality. But the important thing to see here is this line keeps going up, which means the fishing mortality, the number of fish that we're killing, has been going up steadily. So there's two things wrong with striped bass right now. They are overfished with overfishing occurring, which means there has to be management changes to address that. Legally, there has to be management changes. New for last year, a slot limit was put in place where any fish under 28 inches and any fish over 35 inches must be released. My wife was very proud, caught this nice fish, had to go back. This client of mine, also very proud, caught a very nice fish, had to go back. It was too big. And so basically, the point of having a slot is to allow these small fish to get big enough to spawn while still protecting the biggest fish, which produce the most eggs. Very similar to our lobster management in Maine, where we have both an undersized measure and an oversized measure, to let a number of fish or a number of individuals get to spawning size, have a couple of years where they are vulnerable to fishing, and then after that, they're protected for the rest of their days. So based on this graph of the fishing basically getting worse around here, in 2013, Coast Conservation Association of Maine came to GMRI and said, listen, the fishing is declining here. What can we do about it? We said, well, let's think about it. And so we came up with this collaborative project called Snap-a-Striper. And so the idea was to figure out if the fishing in Maine was getting worse, and we're so reliant on, or we think we're so reliant on these migratory fish in the Chesapeake, is there anything we can do management-wise that will protect our fishery? Or are we completely at the whim of the Chesapeake fish? And so, as I mentioned earlier, there is a spawning stock biomass in the Kennebec River. We don't know how big it is. We don't know much about it. And so the central question to Snap-a-Striper is trying to figure out where the fish we catch in Maine, where they come from. We know that most of them are migratory, but we don't know how many of them are local. Like I said, the Kennebec River has special regulations to protect spawning fish, and kind of asking, how necessary is that, or how important is that? So there's two ways we're going about this. We've been getting fishermen to take pictures of their catch and release fish. This is really novel for fishery scientists. Normally, we kill things. That's as simple as that. That's how we study things. We get a lot more information from fish when we can dissect them and take tissue samples and all these things. And so doing catch and release science is really new for a lot of us. So what we're doing is we're doing body morphometrics. And so we're taking photos of the fish, and we're drawing these measurements that describes the shape of the fish. And that's important because the difference in a fish's body shape actually can be indicative of the stock that it's part of, the spawning stock. So on a really simplified way, the difference between basketballs and torpedoes. Oftentimes, across species of fish, fish that have deeper body, shorter heads, they don't migrate as much. Or it's indicative of that fish not migrating itself very much. Think of a couch potato versus a marathon runner. The torpedoes are generally longer heads, shallow bodies, more streamlined fish. And this is not due to genetics. This is really due to life history and how the fish, individual fish, how they live. And so what's really important about this, like I mentioned earlier, the Kennebec River fish are genetically the same as Hudson fish because they were all brought the seed to start the Kennebec population was from the Hudson River. So they are the same genetically. So doing a genetic study, you probably won't be able to see any differences between Hudson River fish and Kennebec River fish. So we're doing these other methods to discriminate the differences between them. Another thing we're doing is otolith chemistry. And so we're getting fishermen to give us the ear bone, or to give us the heads of fish that they legally harvest. The otoliths, which is Greek for ear stone, are the inner ear of the fish. And they grow rings just like a tree. As you can see in this picture, this is a cross section. This fish is 1, 2, 3, 4, 5, 6 years old. What's really cool about this is not only can we age the fish, but we can also drill out the middle of that ear bone. That's the core of the otolith. That's the oldest part of the fish, and it's metabolically inert, which means that that's a time capsule to the fish's first year of life that never changes. And so we're doing stable isotope analysis that looks at the chemistry, the water chemistry that fish is swimming in. And so we can look at the water chemistry to help determine which river system fish had come from as juveniles. So to date, we've received over 2,000 photos of catch and release fish up and down the coast, primarily around Casco Bay and Saco Bay. We've also collected about 80 pairs of otoliths. So some of the things that we've learned, the size of the fish, in general, of those 2,000 fish, typically they're quite small, these schoolies of sub 20 inches, 17 to 20 inches. Some of our bigger samples have come from Saco Bay and south, basically south of Biddeford. We can look at the age of the fish in our sample size, where most of the fish, so remember, most of the fish that we get we get the ear bones for are legally harvested fish, which means at least 28 inches. So most fish in Maine, once they become legal size, is but it's between 6 and 8 years old, generally speaking, to be a 28-inch fish. Over the course of the study, the regulations have changed a little bit, which is why we have some younger fish. Yeah, so we're working on that. The trouble that we have, though, is we don't have many samples. We have very few samples, actually. We have both the picture of the fish and the otolith for the fish. So we've been having a really hard time combining the chemistry data with the shape data, which brings us to kind of the next step, the next iteration of this project we're hoping to do. If we get funding, we're going to call it mail a scale, because we can't help but have cute names for projects. And this will be with collaborators in the University of New England. And it'll be similar in that we'll have fishermen take pictures of the fish that they catch and release, but also take a small scale sample, a couple scales from the backs of the fish, because we can do chemistry, water chemistry on those scales. And so that's non-lethal sampling for chemistry. The data generally aren't as good as with an otolith, especially since you think about a fish can lose scales throughout its life and they regrow. And so if you don't get, you never know if you have the oldest scales on the fish that show that that core area, that origin of the fish. But it should get us the opportunity to get a lot more paired samples of chemistry and morphometrics. And with those paired samples, the idea is that we'll get an idea of what, say, Kennebec fish are shaped like based on the chemistry and the photos. And then we can extrapolate across the rest of our data set, those other 2,000 photos, to make predictions about whether those are Kennebec, Hudson, or Chesapeake fish. So we want to leverage this large data set we already have, and it's really helpful that we get the help of citizen scientists. And so anyone can participate in Snap a Striper. You can send me an email at stripers@GMRI for more information, and also to submit pictures on I forgot to put them up here. On the CCA website, you can print off these cards, these data cards, which are really important to give data for us to be in the picture of where the fish came from, but also for size. Those cards are all 5 inches long, and it helps us to have a scale. But yeah, so you're welcome to reach out to me for more information about this, especially if we get the funding for the scale project. You'll definitely be hearing from me, as we'll try to recruit more fishermen from up and down the coast to help us learn more about the fish that we care so much about in our backyard. There's also a GMRI Ecosystem Investigation Network page that I can send if people are interested to learn more about Snap a Striper. And we've made it together. We're at the end here. So like I said, I've got 2 hats on tonight of being a guide and being a scientist. You're welcome to send me an email anytime to my GMRI email or my personal email. My company is Naturalist Adventures guide service. My website's naturalisadventures.com, and I'd be happy to chat with any of you anytime about fishing. So Yeah, thank you very much, and I'll be happy to take some questions.
Well, someone did ask if this is being recorded, and it is. So we
Oh.
will have that recording up on our website in a
Oh,
couple
good.
of days. And I think we'll probably send it out to everyone in case not everyone made it due to our
Yeah.
late start. And we'll also include any links that you mentioned that aren't in here. We can, edit them into the final video. We did have a couple last questions. One was about, are you finding contaminants or pollution in the fatty tissue of striped bass?
So we haven't done any toxicology work ourselves. Mainly, usually we just get the heads from the fish. When people give us physical samples, a lot of times the PCBs and other nasty chemicals are in the what we call the bloodline on the side of the fish. It's red meat, and so a lot of times we don't have access to that anyway. But no, we haven't done any toxicology work or human health.
Then there's another question here about the balance between, it says, do you have a sense of fish mortality from recreational fishing
Oh,
versus commercial fishing?
so so I don't have that graph up here, but the latest stock assessment shows that recreational fishing accounts for 5 times more mortality than commercial fishing of striped bass. And while that seems counterintuitive, because uh, recreational fishermen catch and release so many more fish, it's because they do catch so many fish. And the estimated mortality is 9%. So it goes into the stock assessments. Of course, you can yourself do better than this, but it goes into the stock assessments that 9% of every fish you catch and release, 9% of those are going to die. And that's due to playing them too long and they get too exhausted. If they swallow hooks, that's very bad. That's why we have rules about what types of hooks you can use if you're bait fishing. So there's something that we can all do to have better practices. Keep fish wet, only take them out of the water briefly to unhook them, maybe take a quick picture. But it's too easy to just blame commercial fishermen for the problems in the striper fishery. That being said, I think everyone has to look in the mirror about how they're handling fish and how we can be doing things better, both on the recreational and commercial side.
Okay, well, we've got some recommendations from some other
Sure.
fisher people here, which maybe you can just comment briefly. Something of the WIS W7T clippers helped save schoolies. Does that ring a bell to
Oh,
you?
no, I don't know that.
Okay. So if people want to go back into the recording, they can read this in the chat from, I think it's John is sending
Oh,
this suggestion. And then we just have lots of thank yous from people about all the great information you've.
well, thank you. I appreciate it.
Oh, and one last question, guide or scientist, which one is your favorite hat?
uh, let's see. With all these GMRI logos on here, I'm not sure which way I'm supposed to answer this. Um, like I've always said, my Latin teacher in high school, his favorite saying was, "Happy is the man whose vocation is his avocation." And so I am very lucky that I get to play with fish all the time, whether that's recreationally, commercially, um, scientifically. I like it all very much, and a big part of why I like fishing so much is the science. And you know, I see the natural world as a giant puzzle, and trying to figure out where fish are, why, and when. So um, they complement each other very well.
Well, that's a good way to end, I think. It's a
All
wonderful
right.
presentation, and thank you for providing your email. If people have further
Of
questions,
course.
maybe they can reach you that way. And
Great. All right. Well,
thanks.
thank you very much.
Thank you. Thank you, everyone, for coming.